My name's Samuel Patrick Naylor. I'm a postdoctoral research fellow here at Oxford University, where I work in the Department of Physiology, Anatomy and Genetics, and I'm very generously funded by the Newfield Medical Fellowship. So my interest in AT probably came about 10 years ago. I was always interested in pursuing a career in cancer research, and so I did a research placement year with Professor Martin Lavin at the Queensland Institute of Medical Research. And so Martin's lab works on AT, but also on understanding the mechanical aspects of DNA damage and how this relates to both cancer and neurodegeneration. And so working in Martin's lab really had a massive impact on me, seeing how hard everyone there worked because they knew what a toll AT took on AT families. And so I then completed a PhD both with Martin and with Professor Ernst Wolvetang at the University of Queensland, where we made the first iPS cells from patients with AT. And when I finished there, we were really only scratching the surface with what we could do with these iPS cells. And so I knew that I needed to come to a lab who were world leaders in cerebellar ataxia, which has led me here to work with Esther Becker in Oxford. Action for AT and BrAshA-T are very generously funding us here to take forward our work on using iPS cells from AT patients to make brain organoids or little mini brains. So we are funded to do this from a handful of Australian and British AT families. So these little mini brains are a few millimeters across, and really this is the most current or the best model of the brain that we can produce as a research tool in the lab. And so the brain is one of the most complicated things that exists in nature, and so we're using new technologies like single cell sequencing, where we can take apart these organoids one cell at a time. We can trap them in an oil bead emulsion, and we can sequence now individual cells from individual organoids. So the way in which we plan to carry out this research is spending a lot of time in the lab, but also a lot of time analysing data and doing computational analysis. So I've spent about a year figuring out how to make these cerebellar organoids, and about a year trying to figure out how to sequence them and analyse this data. And so the last year of this fellowship is really about putting through these iPS cells from AT patients and doing this in a much larger scale. So this involves growing iPS cells in the lab, and then turning them into brain organoids, and then doing a series of validation steps on these organoids, which are biochemical tests, amplifying up genetic material, freezing these organoids and cutting very, very thin sections of them and then looking at them under a fluorescent microscope. But ultimately our endpoint is to sequence individual cells from these organoids and then to analyse that data. So this study is really important to AT patients because what we're doing in making these iPS cells is making a version of the AT brain in the lab. So we're making an organoid that is reflective of that AT patient's whole genetic background and their particular mutation. So there's a spectrum of ways in which AT can manifest, and we can model this spectrum by working with lines from different patients and having really these organoids, which are really models or avatars of a developing cerebellum. So this is really important because drugs have different effects based on people's genetic backgrounds. One drug might work for someone, but not someone else. And so really the more iPS cells and organoids we can make as models for AT, the better chance we have at really understanding the full phenotype of this disease. So in the long term, the difference that we hope to make to people living with AT is really developing a tool that can be used in the fight against AT. So it's a tool that we really hope to learn from, but we also hope that clinicians and other researchers are going to take up this tool and use it to advance their understanding of AT. Now, if you think about testing of drugs, you can't really test drugs in high throughput on patients, but what we've got is, if you've seen the movie Avatar, we've got an avatar that we're sending into battle for you. Your avatar is thousands of these brain organoids that we're growing in the lab that we can test drugs on, that we want to be able to know has an effect to restore the consequence of AT. So we know that pharmaceutical companies aren't really interested in AT because it's so rare. And similarly, it's very hard to get funding for rare diseases like AT. A lot of the funding tends to get given out to more common diseases like Parkinson's and cancer, which really is a bit short-sighted because studying AT actually helps us learn about a lot of these other diseases. The gene mutated in ATM, is so instrumental to so many processes required by the cell that really learning about it is such a beneficial endeavor.